Okay, hello everybody. So my name is Koen Haak, and I'm going to talk a bit about the basic ideas and considerations behind connectopic mapping and spatial statistical modeling. So understanding the fundamental organization of the brain is one of the principal aims of imaging neuroscience. And one popular approach is to parcellate the brain into a mosaic of brain areas with assumed piecewise constant structure and function. This has been extremely useful for understanding how different patches of brain tissue can serve specific functional roles. And it also lies at the foundation of understanding how different functional areas interact with each other. Indeed, by assuming that the brain is composed of a mosaic of brain areas with homogeneous structure and function, one greatly simplifies inference because it enables simple A versus B comparisons, and it also permits outer product models of brain connectivity. Whenever the assumption fails, however, these approaches inevitably lead to inaccurate models of brain organization, and they also conceal local patterns that may be crucial to performing certain neural computations. And the problem is that it's not very difficult to find examples of brain areas that violate the implicit assumption of functional homogeneity, such as, for example, primary visual cortex. Indeed, it is well known that primary visual cortex contains not one, but two overlapping modes of variation. One that represents a distance from fixation in visual space, also known as eccentricity, and one that represents the angle around fixation, also known as polar angle. And this retinotopic organization is preserved across the visual hierarchy, which implies that also connectivity is organized in overlapping modes of spatial variation. Now, retinotopic cortex is certainly not the only part of the brain where connectivity is governed by functional heterogeneity and multiplicity. These topographic features can be found across the brain, suggesting that local connection topographies are not just useful for sensory processing, but also for mechanisms of coding, learning, and even reasoning. Now, besides being fundamental to brain organization, functional heterogeneity and multiplicity are also tremendously important for how we can go about analyzing our data. And to illustrate this point, consider, for instance, this brain region, where one mode of organization varies smoothly in one direction. What exact feature is being described by this map is not very important. It can be either stimulus response, task activation, or connectivity. The same principle will apply to each of these. Consider also that this first mode of organization coexists with another mode of organization, varying smoothly in a perpendicular direction. Our measurements, however, would capture the superposition of these two modes. And this means that if we would try to infer the spatial organization of this region directly from the measurements, we would wrongly conclude that things vary along the diagonal. Even worse, if we would attempt to parcellate this region into two subregions, we would end up with two parcels that are clearly nonsensical with regard to the true underlying modes of organization. What we should do instead is a spatial variance decomposition, such that our measurements are decomposed into modes of variation that are biologically meaningful. Now, to achieve such a spatial variance decomposition, we recently proposed the Connectopic mapping framework. It constructs a similarity matrix of all voxel or vertex pairs within a predefined region of interest, based on their resting state functional connectivity. This similarity matrix is then submitted to a nonlinear manifold learning algorithm called Laplacian eigenmaps, which decomposes the connectivity fingerprints into multiple overlapping modes of variation, called connectopic maps. Now, conceptually, this approach is very similar to the more widely applied variance decomposition method known as principal component analysis, or PCA. However, unlike PCA, nonlinear methods can deal with data that are nonlinearly embedded in a higher dimensional space, such as in this example, where a two-dimensional manifold is embedded in a three-dimensional space. Linear methods, like PCA, cannot capture the local structure of the manifold, and will indicate that the principal modes of variation follow the diameter and width of the Swiss roll. Nonlinear methods, such as Laplacian eigenmaps, on the other hand, try to capture the local structure of the manifold by first representing the data as a graph with connection only between nearby data points. And this graph can then be unfolded by performing a variance decomposition of the graph instead of the data itself. Using this approach, we were able to isolate a mode of variation in motor cortex that resembles its somatotopic organization, as well as multiple overlapping modes in primary visual cortex, resembling that region's eccentricity and polar angle organization. In other words, we were able to validate the proposed framework in regions where we have ground truth. However, one of the more interesting applications of connectopic mapping is to learn something about the functional organization of brain areas where we do not have such ground truth. For instance, to be able to account for functional heterogeneity and multiplicity in brain network modeling, we might want to learn if there are meaningful modes of spatial variation within certain multimodal association areas. In such cases, validation can be achieved by determining whether individual variations in the connectopic maps are related to individual differences in other traits, such as behavior. Therefore, we also proposed an approach for performing statistical inference over the connectopic maps. We borrowed this technique from the geosciences, and in contrast to statistical tests that are common in our field, such as detecting activation or co-activation, it involves a multivariate spatial statistical approach that enables performing inference over the topology of the maps as a whole. To illustrate how this works, let's say that we've successfully extracted one connectopic map. This map may exhibit all kinds of curvature and can be noisy, particularly at the single subject level. But the key idea is that we fit a model to this map by estimating parameters gamma. In our work so far, the model involves a polynomial basis set, but this can in principle be any basis set that is deemed appropriate. And once the model is fitted, the parameters will yield a prediction of the map, which can be compared against the map itself to determine the goodness of fit. Because we use Bayesian regression as a model fitting procedure, we can also compute the likelihood of the model given the data, and thereby determine how many polynomials are necessary to model the map accurately with as few parameters as possible. Now, this approach is not just applicable to modeling spatial maps on a cortical sheet, because it can also be adapted to deal with subcortical structures that are three-dimensional by nature. Finally, because we have just characterized the map in terms of just a few parameters, we can now test whether the topology as captured by the parameters co-varies with behavior. We have now applied the connectopic mapping framework in a number of brain areas and been able to link the spatial organization of these areas to behavior. The general approach in these studies was simple. We applied connectopic mapping to the resting state fMRI data of the Human Connectome project and then used the spatial statistics approach to obtain for each HCP subject a limited set of connectopic mapping parameters, or CMPs. Next, we leveraged the fact that the HCP dataset also contains many behavioral variables for the same subjects, which we linked to the CMPs by standard statistical modeling procedures, such as multiple regression and canonical correlation analysis. We and others have now applied this pipeline in a number of brain areas. One example is particularly worth highlighting because it underscores many of the points that were made so far, and it also follows the recommended procedural steps outlined on this slide. The work considered the functional organization of human striatum, and we applied connectopic mapping and spatial statistical modeling to the subcortical nucleus in the basal ganglia. Striatum is a critical component in the brain's motor and reward system, and therefore of great interest in investigations into neurological and also psychiatric conditions such as Parkinson's disease and addiction. And our work was motivated by the observation that existing functional connectivity studies into human striatum often ended up with a subparcellation that did not correspond very well to characterizations of striatal organization in experimental animals. Indeed, the animal studies consistently described the presence of a rostrocaudal gradient relating to both nigrostriatal and corticostriatal connectivity. The human work, on the other hand, described the striatum in terms of subdivisions that appeared to follow a combination of the striatum's anatomical constituents, that is, the caudate nucleus, putamen, and nucleus accumbens, and a discretized version of the connectivity gradient that has been described in the animal literature. In the context of multiple overlapping gradients, this suggested to us that the human functional connectivity studies had failed to account for the functional heterogeneity and multiplicity of the striatum. We thus applied connectopic mapping to the striatum, and the results confirmed our expectation that the striatal connectivity patterns observed in previous work were actually manifestations of multiple superimposed patterns of connectivity. The first pattern showed rather sharp transitions that followed the distinction between the caudate nucleus, putamen, and nucleus accumbens. The second reproduced the rostrocaudal corticostriatal connectivity gradient predicted in the animal literature. And when we employed spatial statistical modeling to summarize this gradient in terms of 9 model parameters, that is a cubic model in 3 spatial dimensions, and submitted these parameters to a canonical correlation analysis, or CCA, we found that individual variations in the rostrocaudal connectivity gradient are related to individual variations in goal-directed behavior, such as delay discounting. In recent still unpublished work, we additionally found the presence of a third overlapping gradient, which showed a striking resemblance to the spatial patterns revealed by that SPECT imaging, and whose spatial model parameters could be related to dopaminergic mechanisms, such as disease severity in PD, sensitivity to L-DOPA, and the amount of tobacco and alcohol usage in healthy HCP participants. Thus, the striatum is an example of a brain area that exhibits meaningful functional heterogeneity and multiplicity that should be considered before averaging signals from across the striatum or in the context of network modeling studies. In addition, we believe that by decomposing striatal organization into biologically meaningful components, we improved our ability to probing specific aspects of striatal functioning. Now, besides testing whether the estimated gradients express meaningful variance related to behavior, experimental conditions, or group differences, the spatial statistical modeling also enables testing hypotheses about the spatial organization of connectivity within brain areas. And we do this by performing model comparisons. In particular, it enables statistically principled comparisons between different models of spatial organization, such as smooth versus sharp transitions. And this means that it can be used to determine whether the data provide evidence of area boundaries or not. Now, for example, say we're given an estimated gradient in some region of interest encompassing two biological areas. If the two areas have clearly distinct features, that is, connectivity fingerprints, and if the imaging data is sufficiently precise, the estimated gradient will exhibit a rapid transition at the area boundary, such as, for example, was the case in the first connectopic mapping human striatum. We can now use spatial modeling and alternative hypothesis testing to determine whether the gradient can be taken as evidence of an area boundary by comparing the fits of models of smooth basis functions against models with sharp, that is, stepwise basis functions. Smooth models, such as those based on polynomial or Fourier basis expansions, will require a much greater model complexity, that is, many bases with non-zero weights than models that allow for step functions to model rapid transitions accurately. So to summarize, we argued that the assumption of piecewise constant brain areas is generally violated, and that connectopic mapping and spatial statistical modeling are tools to start characterizing the functional heterogeneity and multiplicity of these brain areas. So where do we go from here? Well, in the first instance, we wish to resolve a number of loose ends. So first is the question of how many higher order gradients are actually meaningful. And this is in part an empirical question that can be answered on a case-by-case basis by assessing a gradient's or a connectopic map's ability to explain behavioral variables. Generally speaking, however, it is a problem of intrinsic dimensionality estimation, which is currently an unsolved problem in the manifold learning literature. A related open problem pertains to the fact that most decompositions lead to orthogonal representations, leading in turn to decreased degrees of freedom for the estimation of higher order gradients, and thereby decreased likelihood that they too are meaningful characterizations of behavior. Second, while connectopic mapping allows for eliciting and modeling the spatial pattern of functional heterogeneity, it does not quantify the amount of heterogeneity. Developing quantitative measures of the amount of functional heterogeneity could be important to determine whether it be reasonable to summarize signals into a single estimate, or to determine if modeling the internal structure of a region is worthwhile at all. It will also be important to integrate connectopic mapping and region parcellation into a single framework. Although the connectopic mapping procedures are relatively robust to small differences in region definition, inaccuracies in definition can affect the results. And the most pronounced differences in results can be expected when one region definition does and another does not include patches of cortex with very different connectivity patterns. If included, most of the variance can be explained by the very different connectivity patterns, yielding gradients or maps with very rapid transitions. And if they are not included, most of the variance can be explained by more subtle and gradual modes of variation. Now, these features can be quantified using spatial statistical modeling, and it is therefore conceivable that region definition and gradient estimation, or connectopic mapping, are iteratively refined in a single automated estimation procedure. A fourth important direction for future work is to integrate connectopic mapping in brain network estimation. In the face of functional heterogeneity and multiplicity, accurate network modeling requires accounting for the complex hierarchical covariance structure within and between areas. Connectopic mapping and spatial statistical modeling methods provide a means to understanding this structure, but have yet to be incorporated in a framework for network estimation. Finally, it's worth noting that connectopic mapping may lead to new methods for learning in what way the spatial organization of specific brain areas supports specific cognitive functions. For many brain areas, it's still unclear what functions they support, let alone how we should vary an experimental stimulus or task to evoke responses at specific topographical locations. By first employing connectopic mapping, we can derive possible spatial dimensions along which we can expect functional differentiation, and this in turn can be exploited to reverse engineer that area's functional properties. Okay, so this is all I have. I would like to thank my collaborators as well as the funders of this work. If you'd like to read a bit more about this topic, please check out our latest paper, which will appear in the Gradients of Brain Organization special issue in Mirror Image. Thank you so much.